get started. Did we make it? Oh, we need to start penalizing ourselves for not being on time. This isn't my responsibility, right? It's not like I packed the schedule too tight or anything. I'm actually very impressed. We're only 2 minutes behind. So we have another fabulous panel here today. Because we were running late, because I thought we were having just a great session with Nicole this morning, a couple of the panelists who I knew were fairly busy today. I actually talked to them and asked them if they would mind not coming, because I definitely wanted to make time for this panel, and as you can see, we're running behind, and I didn't want to eat too much into your lunchtime. No pun intended there. So we're going to start now. It's the same format that we've been doing all week. We're going to have 5 minutes with each of our fabulous panelists, and then we will do a Q&A, all right? And we're just going to jump right in and get started with John Snowden, if you'd like to come up. John is, well, he'll tell you about himself. He's an epidemiologist up at OHSU and has some really fantastic research going on, and I will get out of the way and let you start.
Great, thank you, Dawn. So as Dawn said, I'm an epidemiologist and health services researcher in the OBGYN department at OHSU. And today I'm going to be talking to you about my research on addressing health disparities using epidemiology and health services research. So my motivation as a researcher is to address and to tackle health disparities. Healthy People 2020 defines health disparities thusly. Health disparities adversely affect groups of people who have systematically experienced greater obstacles to health based on their racial or ethnic group, religion, socioeconomic status, gender, age, mental health, cognitive, sensory, or physical disability, sexual orientation or gender identity, geographic location, or other characteristics historically linked to discrimination or exclusion. So I like this definition because it really foregrounds the historical and political dimensions of health disparities. And in our society, disparities are everywhere, unfortunately. And I would argue that as health researchers, it's not enough to improve population health. We also have to close health disparities with our research. So to put this in context of my own research, I'll talk today about disparities in maternal death. So maternal death was common before the 20th century, and it decreased more than 50-fold in the 20th century. So this is really one of the signature accomplishments of medicine and public health. Troublingly, in the last 20 years, the rate of maternal death has increased. It's almost doubled. And also troublingly, there are racial disparities in this catastrophic outcome, with African American women more than 4 times as likely as their white counterparts to die during or after childbirth. So this is an unacceptable disparity because it represents both an issue for women's health. Of course, men are not at risk for this outcome. And there's also a very pressing racial disparity here. So in terms of the tools that we have to address health disparities, there are many of them. We can talk about advocacy, activism, public policy, government, business and more market-driven solutions, and finally, my favorite, research. I would also argue that research is a tool used by practitioners of each of these fields to address health disparities. So it's really the foundation. So I'm an epidemiologist. Epidemiology is the study of the distribution and determinants of disease and health-related states and populations. And I also do health services research, which is the study of how people access healthcare and the costs and outcomes of that care. So I'm going to talk some more about my research and disparities in HIV. The HIV epidemic is particularly acute among Black gay men, with HIV prevalence as high as 20 to 30% in some US cities. So this represents, again, a real double disparity because we know that African Americans are at a higher risk for HIV acquisition, as are gay and bisexual men. And so Black gay men really are at the nexus of this disparity. And so the question becomes why, and how do we address this, right? So with colleagues at San Francisco Department of Public Health, I've done research on factors like sexual and social networks, which is what this graphic here shows. Also issues like access to healthcare and discrimination, which in part explain these disparities. I want to note here that it's not necessarily individual choices that contribute to these disparities. And in fact, when surveyed, Black gay men report lower rates of risk factors for HIV acquisition as compared to their white male counterparts. So bring it back to maternal health and this issue of Black women having higher rates of maternal death. We also see higher rates of other conditions among Black women, including hypertension, obesity, factors such as poverty, and lastly, C-section, or cesarean delivery, which is a focus of my research. And that's what this picture here shows. And C-section is itself a risk in the future and future pregnancies for maternal morbidity and maternal mortality. And other contributors that we can think about for this disparity that we see in maternal mortality include access to healthcare and insurance and discrimination. So in conclusion, I've highlighted some persistent health disparities we have in our society, and I've argued that we have to consider factors at the population level as well as the individual level. And that is really the goal of epidemiology and health services research. And finally, once we've done the research, it's important to determine the solutions to put our findings into practice. So I highlight two examples here. One of them, in the case of HIV, is treatment as prevention, or pre-exposure prophylaxis, PrEP, which has gotten a lot of media attention recently. And this is based on research showing that HIV drugs, antiretrovirals, are actually very efficacious as prevention for HIV as well. And this is really changing the landscape of HIV prevention. And in the case of maternal death, there's been a lot of research in recent years on postpartum hemorrhage, which is an acute blood loss after birth. And we now know that postpartum hemorrhage is one of the leading causes of preventable maternal death. And so there are approaches at the hospital and clinician level to address postpartum hemorrhage, things like checklists and protocols, with the aim of decreasing maternal death and also hopefully decreasing this disparity in maternal death. And so with that, I'll close by saying, let's conduct rigorous research to close these health disparities. Thank you.
Presentation. Right. That was awesome. Bar, hi. Adrian, you are next. It's hard to be perfect. Here we go.
All right. So my name is Adrian. I am a PhD candidate here at Portland State in biology, and I am working on chemical ecology, which I'm going to shatter the glass for you on how awesome the chemical interactions that plants and bugs and microbes all have in plants protecting themselves. So I'm going to tell you about how and when plants defend themselves, specifically within the context of plants that have a symbiotic relationship with these bacteria that provide nitrogen to them and help them grow. These are called rhizobia. Rhizobia fix nitrogen from the atmosphere. They give it to the plant gives them sugars, which we know that's helpful. It's an agricultural technique, but it also affects the rest of the plant's physiology, changing the way they protect themselves. Why do we care about plant defenses? Well, the choices that we make about what we eat also depends on plant defenses. So whether you're trying to get some antioxidants or you're making your food spicier, some caffeine, all of these compounds, does anyone know what this bottom corner is? Pop quiz. Take a guess. What's that? Someone say it. It's wasabi. Fun. So wasabi, also a plant defense. Nice beverages if you're old enough. And if you need to end the Hunger Games, plant defenses are always responsible. So not only do we make choices about the foods that we consume, but other organisms do as well, which is why plant defense strategies among specific plants can make a difference. Lima bean plants are our current test subject because they have multiple defenses at the same time. This allows me to look at how defending themselves against fungi, as you see in the top corner, or aphids, that's a little aphid leg trapped by hairs, or how plants defend themselves using toxic chemicals such as cyanide, cyanogenesis, or by attracting bugs to do their dirty work, such as releasing chemicals into the air which attract wasps, which lay their eggs into the caterpillars, which then kill the herbivore and protect the plant. Or another defense that lima bean has is extrafloral nectar, secreting the sugary sweet reward to attract ants, which gang up on the plant's enemies. That's called extrafloral nectar. So specifically within this system, I wanted to know how does that last defense, the extrafloral nectar, impact, or how is it impacted by this symbiotic relationship with bacteria below ground? Which one key piece of information is that all of these defenses are expressed differently. So on this tree, you see multiple different lima bean genotypes. We've cultivated it for years, and they have either high or low expression of these traits. And so within one plant, you can see that the extrafloral nectar changes depending on whether or not they have this bacteria below ground. So in this case, there's a negative relationship between plants having this symbiotic relationship and plants protecting themselves by attracting ants, which we followed up with by actually counting ants and found that plants with bacteria had fewer ants. Cool, so what? Why does it matter? Well, what that means is not only are bugs and everything else and us making choices about our food consumption and food webs, but microbes can also be altering that entire food web flow. So a lot of my research is looking at how the microbes are playing into those interacting defense strategies and the above ground food webs. Thank you so much.
All right, Ginny.
So my name is Ginny Garcia Alexander. I'm in the Department of Sociology here at PSU. And I thought I'd give a little bit of background about where I came from, where I got my training. So I actually have my PhD in sociology, but my area, or the way I was trained, is as a social demographer. So what that means, or what social demographers do, is we study population processes, so birth, death, migration, and how those processes impact. In my case, I look specifically at health. So my first appointment was at the University of Texas at San Antonio, and then this job came up with an emphasis on health disparities. So of course I jumped at it and came here in 2012. So very broadly speaking, my research interests are in the area of health disparities. I look at how health outcomes vary across certain subgroups. So I look at racial and ethnic differences, at how, again, processes like immigration impact various health outcomes. So I have sort of my three major research questions or areas. So how social processes, including migration and marriage, impact and produce social and health inequalities, racial and ethnic and class-based disparities in both health status and behavior. And then I also have a stream of research where I look specifically at how religion influences both health behaviors, and then I look at its effects on infant mortality. So these are some of the more sort of specific studies that I'm engaged in, either independently or via collaboration. So I have looked more recently at sort of what's happening with respect to interracial marriages, why that's changing in recent years. I'm also really interested in how the reproductive decision-making process occurs, and more specifically, how sterilizations are administered unevenly. Since I've gotten here to PSU, I've done a lot of work with the Oregon Health Authority. I've been looking specifically at health disparities in the Medicaid population using their claims data. I've also worked with some of the people over at the Center for Outcomes Research and Education. We're doing some studies of with the Oregon Health Plan and what impacts enrollment. We're also looking at quality of care. And then again, I have some ongoing collaborations with people from UTSA where we're looking at bariatric surgery, and we're actually getting ready to put in a project where we will ask for some funding to look at long-term outcomes in people who undergo bariatric surgery. Finally, as I said, I do some work with religion. So there's really sort of quite a breadth of research in sociology, or as a sociologist, and more specifically as a social demographer. So that's it for me.
So far the trend is you've been 30 seconds shorter each, so you can only be like 2.5 minutes.
Perfect. I talk fast.
All right, Ken, you are next.
Great.
I usually start this talk with, and now for something completely, totally, and utterly different, but we're also different. It doesn't really make that much difference here. So I've been at PSU since 2001. I actually moved in on 9/11, so it gives you a bit of an idea when I've been here. I've had about 50 undergraduate students working in my lab on very basic research. I'm going to give you, in the next, what I said, 2 minutes and 15 seconds, and real sort of I don't know, stratospheric view of 3 kind of research projects that are going on in my lab. And that reminds me to mention that I'm part of the Center for Life in Extreme Environments, which is a really globally known research group based here at Portland State, looking at life under the extremes of temperature, pH, basically you name it. I also am a member of the Oregon Nanosciences and Microtechnologies Institute. I'm the virus guy, really little stuff. I'm also an affiliate professor at OHSU, and I started a company in December. So most people, however, know me as the virus hunter. That's what I do for a living. This is me back in Yellowstone National Park. Basically, we poke around hot springs and call it research. But the main message here is actually we're looking for, and I love this is a poster, something you may have seen around campus, finding clues to life in the strangest places. So my background is in chemical engineering first, and then in molecular biology. I'm interested in understanding really little things and then applying them to much bigger pictures. So I'll give you 3 really quick stories about this. And the first one has to do with how old are viruses. So I had a PhD student at my lab and a couple undergraduates working on this as well, trying to find if we could find virus fossils. Why virus fossils? Nobody knows how old viruses are. If we're going to understand viral disease, we should probably get an idea how old some of these viruses are, how they've been interacting with their hosts, probably for billions of years. But we have no direct evidence for that. Are there virus fossils? So what we did is we actually made some sort of fossils in the lab. These are actually some pictures of one of the hot springs we worked with in the background. And these are some bacterial viruses, a plant virus, and some of our favorite viruses, coated in silica, which is sort of the first step on the way to becoming fossils. Unfortunately, these things became blobs, which was not very good. That's why I know missuspected virus fossils. But what my graduate student noticed is actually they're not fossils, they're zombies. And what do I mean by this? Well, it turns out that this fossilization process actually preserves the viruses. And preserving those viruses, now we just showed it for bacterial viruses, but we're hoping to show it for vaccines, which are a huge problem in terms of getting that last mile out into particularly the developing world, but even the developed world. So this is basically the company which is trying to work on this virus fossilization, and we're doing some basic research in my lab about that. The next thing that we're working on has to do with my favorite virus. This is the SSV virus. Those of you who had virology will go, what the heck is that? It's not a giant lime or a little football. It really is a virus, and it's one of those things that you find very commonly in hot spring environments. This particular image right here is taken from about 900 different virus particles that we've imaged over here in the basement of SB1 at our Center for Electron Microscopy. Average all this together, get this really fascinating structure. We think this has to do with the cells are infecting and how it can be functioning under the really high temperatures and low pHs. This guy infects organisms that grow at 80 degrees Celsius and pH 3, basically in boiling acid. How the heck can they do that? That's what we're trying to figure out. But that's just one application of what we're doing, very basic science, but it turns out that this funding, which is funded by NSF, might actually be a way that we can now make nanoparticles, because viruses really are nature's nanoparticles, and use those now for biomedical imaging. It's a long way off, but this is the next step that we really want to take as far as this process is concerned. Then the last project I wanted to talk about is our favorite field site. So again, I tell you about going to hot springs and calling it work. This is literally where we go every year and collect samples. This is in Lassen Volcanic National Park. We've been funded there for about the last 10 years through the National Science Foundation with a group at Humboldt State and Chico State. They're looking at the microbes which are in there. We're, of course, looking at the viruses. The mechanical engineers here actually designed this little boat over here, remote-controlled vessel for going out on the lake. Nobody wanted to go out in the lake to collect samples because it's 50 degrees Celsius and 95 in pH of 2. So not a very nice environment to go out in, so that's how we get this sample. One of the things we found there was this really kind of mind-blowing virus, and actually not really a virus, it's a genome of a virus, and I'm happy to talk more about that later. But the virus kind of looks like this. You know, head of a lion, tail of a serpent, what the heck is going on there? It looks like it's been a fusion between two different kinds of viruses which have come together and are now actually replicating as a particular virus. We're trying to figure out how the heck that took place. And of course, this is important for newly emerging diseases. Hopefully not quite as bad as that one. Well, this is a really excellent movie, and I happen to know one of the advisors as far as putting this whole film together. So that's what we do, a little bit. Sorry, I took more than that.
That was.
Can I use the leftover time?
No, you can't.
Yeah.
You already squeezed your way onto
I
the
know,
panel.
I know,
Come
I know.
on.
Okay,
Here we
sorry.
go.
That's when you put him last, right?
He'll be speaking for the next 30 minutes.
Yes. Okay. Thanks for listening, and thanks, Dawn, for squeezing me to the panel. Disclaimer at the start, just like yesterday there was a speaker on the panel who said they don't have an active research program, well, either do I. But I thought it would be good to present what I've done in the past. And I'm sort of keeping a bit of research going, even though I'm an instructor at the moment. And there's a possibility that some of this can fit into some of the research learning communities that are coming up. So we'll see how that goes. Anyway, first of all, I'm glad that some of the people are talking about microbes, because this building here has got foundations, right? If we knock the foundations out, the building would just fall over, right? That's what microbes are to Earth, all right? So they're sort of like the base level of everything that happens on the planet, the ecosystems, etc. So without them, other things wouldn't happen, okay? So I've been interested in these for quite a few years. But before I do, I thought I'd give you a little bit of background. Some of the speakers told you on how they got to where they are. And so it was good that today we were doing some of that thing about the Jabari square, I think, or the window. And my open quadrant, some of you are probably already forming an opinion of me, right, based on my accent and things like that. So I just wanted to point out that, okay, I was born in Rome because of my name, and yes, I was raised by kangaroos. Okay, and every weekend we just sit in the front yard, put a shrimp on the barbie, have a beer, and watch the kangaroos box. Okay. So I don't know how many of you are thinking about that about me, but that one of those facts is true. Okay. Okay. So, but how I got here, I won't go through all of this, but basically since 1984 is when I started my Bachelor of Science in Australia. And then I did research. I didn't want to do a PhD straight away. Okay. So even though this program is designed for that, you know, if you guys go through the program and you think, I want to take a 1-year break or a half-year break before I do my graduate program, you can still end up somewhere like I did, okay, which may or may not be good, depends on your opinion. But, you know, don't feel as though you have to, bam, go through this. I hope that people who have EXITO are not saying, no, go straight to PhD. So I did do a PhD there, and then I came to do a postdoc up here in 97, and it was in medical genetics, right, so nothing to do with microbiology. Then I went back, got married, and in Australia at Macquarie University I did a bunch of research in a number of different fields, including microbiology. And then came back to America for about 6 years, worked at OHSU, not up here, but there's a campus in Hillsborough, which is sort of half an hour from here, or about 2 hours, 50 minutes in traffic. And someone's already used that joke, I'm sorry. But, and then I'm fortunate enough to be a tenure track instructor at Clark College, where I teach microbiology. The reason I've done this is that basically this is where I just finally found out what my passion was in research, right? I'd been doing research and I loved it, but I didn't quite know. And I found out about astrobiology, which Ken is involved in. I don't know if you use that word, but it's basically, I'll briefly go through it in a minute if I have time. Okay. And then even further, this is where I got into astrobiology. So only the last few bits and pieces. So actually, I'm going to skip this one because I don't have time, so don't worry. So let me just talk a little bit about extremophiles, which Ken talked about. And essentially, they're organisms that can live in conditions that we don't consider normal. So anything that's not like we can live in, okay? So there's some of the examples. And I've been fortunate enough, like Ken said, the reason we do research is so we can go to places like the middle of the desert in Australia, volcanoes in New Zealand, Mount Hood, Antarctica. So that's research, right? And then we come back and study it. So astrobiology, which is essentially the sort of research I like to keep doing through this program, is essentially addressing really big questions, like how does life on Earth begin? How did it start? Okay, how did it evolve? Are there microbes on Mars, etc.? It doesn't study this, okay? So I used this a long time ago. It doesn't work as much now that it's passed away. But astrobiology is very interdisciplinary, which is really important for you guys, all right? So the important thing is that don't limit yourselves to just one field, all right? Get input from everyone, and astrobiology is perfect for that. So the actual project, or areas I'm interested in, is this hot spring. This one's in Australia, but the fascinating thing about this is it's actually radioactive, right? So see those bubbles there? That's radon bubbling up from the bottom. So there's a whole bunch of radioactivity there. And despite that, microbes are growing like crazy, like these cyanobacteria. This is fluorescent imaging. That's my timer, shush. And the things I'm really interested in is I've already looked at what lives there, all right, so that's sort of done. We don't know what viruses are there. We don't know, there are implications for DNA damage repair mechanisms, so how do microbes that are exposed to radiation and all these insults, how do they actually survive, right? Their DNA is either being damaged or they're somehow stopping it from being damaged. And I'm fascinated by how the microbes have evolved into resisting those sort of conditions. Okay, so hopefully something will come up in one of the learning communities where we can sort of work on that. Okay.
So another diverse and fantastic panel. Let's go ahead and do some questions. Yeah, go ahead, Daddy.
This one's for Ken.
Yeah.
Can you just really quickly explain kind of how you use viruses for biomedical imaging?
So can I explain how I use viruses for biomedical imaging? So the idea here, now, as I hopefully mentioned, this is a ways off here, but basically using nanoparticles, what we should be able to do is load them with various different heavy metals, so contrast agents, basically, which we can then, once we understand how to get the metals in, but maybe even be able to use, you saw the little tail fibers at the bottom of the virus, those are the targeting mechanism. So hopefully we can now target, say, a liver cell or a cancer cell or something like that, bring this contrast agent to them. As we all know, we're mostly water. And so the big problem with medical imaging is getting that high contrast, particularly early on in disease, when you can actually do something about it.
Garcia?
This is for Adrian. So you talked about the external floral nectar as being a defense for plants. Is it also possible that predators are also attracted to that, or is it just specifically organisms that are attacked?
Yeah, great question. Is this on? Good. Awesome. Great question. So providing a sugar source, just liquid sugar, is going to attract everything, right? Microbes, bugs that the plant doesn't want to eat it. There's a lot of research actually that study the chemistry of the nectar to figure out and have figured out that there are some like small amounts of toxins in it in case an ant decides, oh, I'm just going to go and hang out by a nectary all day and just drink it all up and not help the plant. But that way it accumulates in the system and is actually toxic if they eat too much of it of certain kinds of plants. So there have been defenses that have evolved in response to that. There's everything you can think of, really. Good question.
Question is also for Ken. What mechanism do bacteria use to survive fossilization, or what kind of environment does that fossilization provide for bacteria?
Yeah, so I don't know how many people got the question, but I'll sort of repeat it real quick. So basically, how do bacteria, for that matter, the viruses, survive under these fossilizing conditions? And the short answer is we don't know, but there certainly are some mechanisms. Best understood, in fact, for bacteria, some of the things that Roberto's been working on, where they have their outer layer, which they slough off, so that gets put in silica, which is what happens in these springs, and then just gets thrown off. How the viruses deal with that is a lot more of an interesting question. It turns out that the actual molecular structure on the outside of the virus determines how much or how little of that mineral they get on the outside. And so it's probably an evolution to give you the hot spraying viruses, which are a lot less susceptible to this, as opposed to some of the other ones which are not.
Ariane?
This question is for Ginny. And what is the most interesting or impactful thing you found on your study of religion's influence on mortality?
So we actually found some really interesting differences by denomination in the studies where we looked specifically at infant mortality. So what we did is we looked at the county level, we looked at infant mortality rates, and then we broke down concentrations of various denominations. So we had sort of mainline Protestant denominations, and then we had a couple of subgroups of conservative Protestants. We had Catholics, and what we found is that having a larger concentration of mainline and Catholic is protective, meaning that the rates of infant mortality are lower in those areas. On the other hand, we found that certain conservative Protestant groups, so Pentecostal, for instance, is associated with a higher rate of infant mortality. So it has some interesting implications for sort of how you can communicate with people about prenatal care and sort of things that can be done in the importance of those sorts of things. So yeah, so some really interesting denominational variations.
Can I ask a follow-up question to that? Sometimes I think, particularly when you share information like that with the population at large, there's an obvious conclusion they draw, which is, oh, being Pentecostal means this, or having Catholic. Can you speak to what these findings actually mean or hint towards?
Yes. So certainly we always have to be very careful, especially in the social sciences, to distinguish between the difference between causality and correlation. So certainly I don't want to paint the picture that being Pentecostal causes higher rates of infant mortality.
I know that, but that's very common.
Right.
That is what most people default to think.
Right, and so all that we found was a correlation, where there are higher concentrations of Pentecostals, and there were a couple of other conservative subgroups that had the same correlation. It just means that there were also higher rates of infant mortality in that area. So we have to be really careful with the way we talk about it, and certainly make sure that we say that we're not making a causal argument there.
And we had Dr. Wallach here yesterday talking about the importance of place and looking at place factors.
Right.
And this is a perfect illustration. We're looking at data at the place level and how that's associated with outcomes across individuals in those places, not causal. But you see these relationships, and it leads to a whole other, there's so many research questions we can think of that stem from that. Yeah, thank you. Yeah, Kim?
Just a follow-up question to that. Were you looking at like Catholics and Pentecostal in similar like urban settings or rural settings, or was it like were there other disparities besides just
We just controlled for those things. So we controlled for urban and rural status. We controlled for percentage of race. We had all sorts of controls that were included. So we didn't necessarily stratify or break it down by other kinds of characteristics, but we did control for those things. So that just means that after considering the effects of something like urban or rural status, there is still a significant meaningful relationship between religion and mortality.
Ken?
So this question is for Ken and Roberto, and I was wondering, so you guys are studying viruses and things that live in very extreme environments. Have you found, or you know of, any that kind of thing in space? Because that's a pretty extreme environment.
Okay, so in the vacuum of space, there's probably nothing. However, there have been experiments done where they put microbes up on like a space station.
Or like on Mars, or like a different planet.
Oh, okay, all right. So,
yeah.
well, we know that they can survive if we put them up. There's some microbes, all right? In terms of on Mars at the moment, probably not on the surface, but there are possibly some areas deep down where there are microbes at the moment. Yeah.
But just wanted to add briefly, this was last week, Monday and Tuesday, I was in Chicago at the Astrobiology Science Conference. And so a lot of the discussions there were about, are we going to find life anywhere else? And all of the current NASA missions have so far been looking for conditions like the ones that Roberto and I are looking at where life could be supported.
What about like fossils of these?
Yeah, so that's fine. That's one of the reasons we're interested in fossil viruses, and that's why we actually got funding from NASA to do this project in the first place. So if we go somewhere else, are we going to find virus fossils? And virus fossils, at least for everything we know about viruses, would be a really good indication of life. And if you look at samples on this planet, if you would take a small sample of soil, of ocean, whatever, you'll find way more viruses in there than anything else, by orders and orders of magnitude, hundreds of thousands of times. So obviously this is one way to look for that. The problem is these things are very rare, and also, as I mentioned, for the virus fossil stuff, they're blobs. And you find blobs everywhere. In fact, the Martian rovers have found blobs. Some of you may have heard that there's a new mission that NASA's going to be sending the moons of Jupiter, probably in the next couple of years. That is going to have some much more life detection-like instrumentation on it. So we're in fact in discussions right now about what instruments to put on that spacecraft to go and look for some of these things. One thing you may have heard about, sorry, monopolizing this here, but places like Europa and certainly some of the other moons of Jupiter, Enceladus, have these huge geysers that shoot off of them. And one of the thoughts is that you could fly spacecraft through these and collect a sample and see what's there.
That's cool. That's awesome. Thanks. Tara.
So over the past few days, this is a general question for the panel. We've heard mention of like collaboration with different like natural sciences. Mm-hmm. I've seen biology and I've seen chemistry. I was wondering, is there a collaboration with physics? And so, if so, could you give examples?
Anybody else? I actually have some very direct examples there. So I have been a co-investigator on a grant with physics funded by the NSF to develop a very specific kind of microscope called a photoelectron microscope. And so we're collaborating very closely on that. Turns out that the particular application, we were looking for viruses, of course, didn't end up looking for that, but it's really helped them move forward with this photoelectron microscope for looking at surfaces. And so there definitely are those kind of collaborations. One of the things that we have at PSU, one of the reasons I'm really attracted to this university is because the collaborations are so good between the different departments. We really aren't in silos here. We really do collaborate very well back and forth. And I think the Center for Life and Extreme Environments is a really good example for that. We have biology and chemistry faculty right next to each other, sharing the same space. Sometimes even sharing students who are between the two different departments.
This is Director Ginny. I wanted to get just a deeper understanding of the study regarding examination of your reproductive distinction and making the sterilization pattern. What goes into that?
So actually, this is a good example of a collaboration because Don worked on this with me. This is a study where we were really interested in looking at in the context of C-section, actually no overlap there, when a sterilization happens at the same time as a C-section. So we wanted to know exactly what predicts that outcome or what makes that outcome more likely. So we used existing data, National Hospital Discharge Survey data, over a period of years to consider the things that make that particular outcome more likely. Certainly we found some racial and ethnic disparity, well, not ethnic because we didn't know if they didn't differentiate between Latino versus white within the white group. But certainly we found racial differences. One of the biggest predictors was Medicaid enrollment. So Medicaid actually accounts for about 65% of all sterilizations that happen in the U.S., which is a pretty startling finding. So that's kind of something that I got together with several people actually over in community health to work with and sort of investigate further, sort of see what was going on there.
Yeah, Shannon and Zach.
I'm not sure, not remember your names, but as you're doing your research, you said that Black women smoke 4 times higher for mother mortality. And so I'm wondering, do you think that's more healthcare, the healthcare that they receive in hospital, or is that exacerbated by pre-existing conditions?
I'll let you piggyback in just, I want to hear John jump in, because this is a big answer that we're about to get here, and so I want to make space for that. Okay, so Zach, we'll come back to you after, okay?
Yeah, that's a great question, and I think that both of those factors are at play here. certainly African American women bring more pre-existing morbidities and conditions to their pregnancies. And as well, we know that rates of appropriate prenatal care utilization are lower in that population, and as well tend to deliver in hospitals that have worse outcomes and lower quality of care. Even at the provider level, there are factors in terms of, there's interesting work now being done on kind of the psychosocial interaction between patient and provider and how that may affect the care that's provided and people's ability to recognize emerging emergencies and everything. And so I think all of those factors contribute to it. And it's a really complicated and fun thing to study just because it is so multifactorial.
Ginny, do you want to? Oh, I was just going to ask on that. Is education a factor? And if it is, in a sense, how what is being done to
to mitigate the risk.
Yeah.
Sorry. Just to say, Larry, when he was here yesterday, Larry Wallach talked a bit about fetal origins a bit, and also talked about social determinants and used this exact example of what we know about African American women with high education, what it means for their infants. So just so you know, they heard this a little bit of that. So.
Okay. And so high education, so when you say education, do you mean like childbirth education, about the childbirth process?
I don't necessarily mean the amount of school that they have, but the childbirth education, and this is a risk. So are we education when we have to be pregnant on this risk and what we can do to make sure that doesn't happen to our children?
Yeah, that's a great question, and I think it's definitely a big factor in this issue that that African American population is less engaged in the healthcare system, and there's historical mistrust of the healthcare system for a good reason, and the quality of care just really is not at that same standard for a bunch of reasons. And that definitely goes to the prenatal period where that kind of patient-provider interaction is providing that education that in part contributes to these outcomes. So I think that's definitely a factor. From my perspective, really an area where more research is needed. Prenatal care is this really old model that we just do, we count the number of visits, and it's not, there's no evidence that we've designed it most effectively. In fact, there really is not evidence based for it. And we could be providing, one of my colleagues at OHSU is in the School of Nursing. She's a nurse midwife and a PhD in nursing. She does really interesting work on group prenatal care models, which is kind of an emerging model that's become more popular in the last 10, 20 years. And so rather than just rushing women in and out of the office for 30-minute visits and taking vitals, the idea is getting groups of 10 or 20 women together and taking vitals, and the midwife usually leads it. So there's a healthcare element, but there also is more of a social interactive element to group prenatal care, so that they can actually share their stories with their previous births and their thoughts and their fears and their expectations for the present pregnancy, and get more of that education that will probably improve that.
And just to piggyback off of that, since this touches on some of the research that I do, it's important to say that certainly the healthcare, where individual women interact with the healthcare system is where a lot of these issues are, absolutely. But even when holding those equal in models and looking at whether or not do they know how to take care of themselves during pregnancy or what to eat or what to do, for example, even holding that, that not being a factor, your own level of education, your actual socioeconomic status is what seems to put African American women at risk within our society. As Larry explained yesterday, right? He talked about the gradient and the hierarchy. We're going to be talking about that tomorrow and Friday. And so particularly when we talk about infant mortality outcomes, and particularly around maternal mortality and other maternal outcomes, it seems to point to the fact that here in the U.S. anyway, experiences of discrimination, racism, and things of that nature are what really are helping to drive some of these disparate outcomes. Does that get to your question too, Shannon?
Yeah.
Okay.
I wanted to just, what do you find as more prevalent? Is it usually the care in hospital or the lack of care? Or does it seem to be more the pre-existing conditions that were unable to be treated?
I didn't hear the first part of what you said.
Alright, so is the lack of the care that they first receive, is the care in hospital that's kind of tends to be kind of more of a problem, or is it the pre-existing conditions that tend to be more prevalent? So is it education for doctors that should be more of a concern, or the access even for the women for better education, better health?
Yeah, I don't want to say both again, but I really do think that it's all factors. I I think that in my research, I tend to focus more on the healthcare system and educating providers and hospital policies, and those are more kind of proximal causes in a short run, and I think that the levers to change those are perhaps more obvious. I would argue, and I think this is sort of tying to what Don was saying, that the more distal causes, the more fundamental causes, these more social causes, which are I think are difficult to study, and really important to study, and we're seeing some really interesting work on social determinants and using really sophisticated methods. Very hard to study, and I think probably more at the root of these issues. But I also think in terms of the lever, how we change them, harder, less immediate to know how we do that. Right. Of course, there are mechanisms and everything, but it's just a harder thing to study with research. But very good.
Right, and trade-offs, lots of trade-offs, yeah, like that.
Actually, this is for Dr. Schnoon as well. Once you've done the research, once you have the information available and are noticing these patterns and trends, how do you specifically turn that into an application? How do you help the situation?
It's a great question.
How do you intervene?
Yeah, and that's something that we're struggling with in public health and epidemiology and policy constantly, and in the last 5 years in particular. And so, I think that it's frustrating to me because it's not something that I was taught in my graduate training. And I think that, we tend to focus on a beautiful statistical model and a new question, and then in terms of translating that into practice or into policy, into patient education, population intervention, I think that part can be really lacking. Would you guys agree?
Yeah.
Yeah. So that's an open question. In my own work, I really try to think about publishing and disseminating the research in venues that are read by and so that it actually reaches practitioners, and so that's so that it's actually used and taken up into practice or hospital policies is another thing that I'm concerned with. But I think those are pretty proximate. I think in terms of how we translate our more population-based and social research into laws, market-based policies, I think that's a really important question.
Yeah. Some of the things Larry was talking about yesterday, a lot harder. And I think as you notice on some of the panels, we have a lot of people who said they do community-based work, community-engaged work, community-based participatory work. When we say community, that might bring something up in your mind, but for those of us who do that type of work, a community could be a community of providers at a hospital. It could be the community right outside the door here. And the reason people tend to do CPPR or engaged work like that is because it lends itself much more, it's a much harder amount of work to do because you're having to develop a lot of trust over many years, as Kelly Gonzalez said. But when you're finally there and you have the research, the people that need to implement that research right away are already there in your camp and they get it and they're ready to move. And that's why, when we think about intervention, I know we talked about this too, Roland, about intervention and what we're going to do, implementation. That's where community engaged work can give us sort of a leg up. Yeah.
Just wanted to add one thought to that. That's an important distinction that happens in all of our research and all research probably on campus, that there's the basic science and then the applied science. So what we see here is the interface between what do we know and what are we going to do about it, which happens in all fields. And that's one of the cool things that Ken was talking about, how he understands that these viruses are able to have silica around them, but then what can we do with this? We can do something about vaccines. So in all research, it's also important that not every outcome, the applied outcomes, aren't always going to be transparent either. So some of these discoveries are so exciting because they look like blue sky research. So funding that kind of research is also really important, but also hard for some of the folks that have the money to understand why you want to go out into a hot spring and poke around, or why do you want to jig up some rhizobia and put it on this plant and see how the bees respond,
Well, I think this goes back to Ken's point about the collaborative nature of what we have here at PSU. I know myself as a faculty member, having been to other institutions before here, I made a very conscious choice to be at a place like PSU versus, a place like, I don't know, for example, University of Michigan, where I did my postdoc. Because there are universities that have certain values, and some of the values that are espoused here are collaboration, working across our disciplinary silos, and you see that. I think you've seen that in the panels. These people aren't paid to be here. I was able to, through my connections and through the connections of the faculty that are in this room, call people from disciplines very different from mine to come and share with you because we have those relationships and we can build on them. And so I feel that being in institutions where you have that ability to talk across these disciplines, it facilitates conversations about basic and applied science and what can we do with this and how can we engage students and all of those things that we're talking about. Let's take just a couple more questions. Yeah, Danielle.
I have a question for Ginny. Have you done any research on like other world religions or just like Christian denominations?
No, so mainly we have focused on the sort of mainline and then mainline Protestant and conservative Protestant distinctions. So really the thing that's shifted has been the outcome itself. So I've done a couple of studies where we look specifically at mortality, but then also some health behaviors, so sort of binge drinking behaviors, smoking behavior, and things like that. But normally there's sort of a pretty standard use of religion in terms of the studies that incorporate various health behaviors, and so we've used that kind of standard model and have not necessarily delved into anything beyond those kind of major ones.
And one last question from someone we haven't heard from. Yeah, John.
This is for both Dr. Roberto and Dr. Ken. I was wondering, what are some of the evolutions you've been able to see inside these practices? What are some of the changes you've been able to see throughout time in these virus fossils?
Speaking to the virus guy here. So I think, to paraphrase your question, what kind of evolutionary processes have we seen in these environments? And to be perfectly honest, we have very little good long-term studies looking at these things. We've done some evolution experiments literally looking at what happens when we make changes. And so a lot of the people in my lab literally do what I could say geneticists do. You break something and then you try and figure out what you broke. And so that process actually has a lot to do then with the evolutionists. And so evolution has basically given us what we have now. Let's see if we can backtrack from what we have now in terms of some of these other things. And that does clearly, at least in our cases for these silica coating, the coatings on the viruses, have very clear indications that the viruses that are present in environments where it would be advantageous to not have this coating happen don't get coated. Those that aren't do. And so there's, again, it's always hard with a time machine to say what's happening with evolution, but you can certainly do some of the things. There are some people that I collaborate with who do what's called experimental evolution, where they literally will take fruit flies or bacteria or viruses and then follow them across many, many generations, actually do evolution and then go back. One of the really cool things about the technology that we have right now in terms of molecular biology is you can literally sequence the whole genome. All of our genome, everybody's genome sequence in here, that'll get done probably in the next 10 years. What do you do with it? That's another question entirely.
We've got time? Okay, so for the hot spring, we haven't done any evolutionary studies, but that's why I find it so fascinating, because we, for example, we've all evolved with ultraviolet light. So we have mechanisms to protect when our DNA gets damaged by ultraviolet light. We have things in our cells that will fix that. Okay. But we haven't involved so much with ionizing radiation, things you get from uranium and things like that. So that's why this hot spring will be so fascinating to see organisms that have evolved in those conditions, to see whether they have any extra mechanisms to protect them, and which has the implications for cancer and things like that, even humans, etc. So we haven't done it, but we'd like to.
So I want to thank our panel. We are about to have lunch. I have invited them as always. And of course, if you run up here after, you're basically going to trap them and you can ask them all kinds of questions.